Hello everyone and welcome back to Human Biology here at Chaminade University. Today we will be discussing genetic inheritance. So we'll be talking about Mendelian inheritance. Gregor Mendel was a monk, an Austrian monk, who utilized the monastery's pea garden. They were using pea plants to feed the monks, and he utilized the peas to be able to investigate mathematically how we might be inheriting what he called inheritance units, which we now know are genes or chromosomal inheritance. So this here is a chromosome before it's been duplicated. Here's the centromere, the short arm, the long arm. Genes within chromosomes are organized in a very specific way, just like books in a library have a specific location. So genes are going to be organized in a particular what we call locus or location within the chromosome. And when we duplicate this chromosome, we're going to have an exact copy. So here's a duplicated chromosome with gene 1, gene 2, and gene 3 in the exact same location. These are called sister chromatids, and these are formed during DNA replication. These are going to be exact copies of one another. Okay, and as we are going to inherit these information from our parents, we will be passing these genetics down in the form of these chromosomes, or what he was calling inheritance packets. Again, he didn't really know what we now know in terms of the field of genetics. But let's talk about some definitions here to get started. So heredity is when we are taking traits and transferring them from parents to offspring. Anything that is heritable is something that is an external trait. An external trait is a phenotype. And a heritable trait just basically means that it's something similar that's passed down from parent to offspring, etc. There's a ton of different examples of human phenotypes, including hair color, eye color, height, etc. And all of them are controlled by our genetics, which are the genes that are found on our chromosomes. So we as humans have 46 chromosomes. We have 23 pairs. Namely, we have 22 autosomal pairs, which gives us 44 autosomes, and 2 what we call sex chromosomes. So one sex chromosome pair that in men is going to be XY, and ladies it's going to be XX. So because we have pairs of chromosomes, that means that we have 2 copies of every chromosome, meaning that we also have 2 copies of every gene. Now, just because we have 2 copies of every gene does not mean that they are identical. They could be the same, but they could also be different. And we have different variations of these genes that are called alleles. So if, for example, the gene calls for eye color, an allelic variation might be blue eye color or brown eye color, etc. And all the combinations of the alleles that we have in our chromosomes make up our genotype. And our genotype, or our genetic composition, is what is going to make up or create our phenotype or our outward composition. Okay, so let's talk about some of the terms that we're going to be using here. So when we talk about genotype means the combination of alleles that you are given from both your maternal and your paternal chromosomes. Now, sometimes these alleles are going to interact in what we call a dominant and a recessive way, and we'll see that in just a second. A really good example of that is purple flowers in the peas. So a purple flower is going to be dominant over a white flower. Why? Because having pigment trumps no pigment, no matter what. So you could have 2 alleles that code for pigment and be purple, or you can have 1 allele that codes for pigment and still be purple. You would need 2 alleles that do not code for pigment in order to be white. So now let's go back to this genotype up here. The genotype indicates here that we have 1 copy of purple pigment and 1 copy that does not have purple pigment, or this would be the white. So this would be considered what we call a heterozygote. In a heterozygote, we do not have exact matching of alleles. We have 2 different allelic variations at a locus, one on the maternal and the paternal chromosome. For example, at eye color location, we have one that's brown eye color and one that's blue eye color. And in this case, brown eye color would be dominant over blue. All right, so I'm going to leap back to this slide here, and we're discussing the way that we are going to be able to take our genotype, which is in our DNA, and express it outwardly, which is what we call our phenotype. And to do that, we need a little connected molecule here called mRNA. So DNA makes RNA, and RNA makes protein. But again, depending on what your DNA says, that's going to make a copy of it into the RNA, and that RNA is going to be making exactly whatever that mRNA says into the protein, which is going to be what the outward appearance of the individual is going to express, namely the phenotype. Okay, as I mentioned, Gregor Mendel was an Austrian monk, and he utilized pea plants from the Austrian garden because they were very frugal, and this way they could utilize them and eat them after he was finished his mathematics. He studied the phenotypes of multiple generations of peas, and specifically, he studied some of the outward appearances, such as purple flowers versus white flowers, which we just talked about, purple being dominant to white, yellow seeds over green seeds, wrinkled seeds or round seeds, and tall or short plants. He has studied many, many more phenotypes than this, but these are just some of the ones that are classically known as the ones that he used. Here's a series of the different types of traits that he looked at. So he looked at seed shape, seed color, pod shape, pod color, and flower color. And then what he did was he would cross these. So he would cross a round with a wrinkled and see what he got. He got 100% in the first generation all round. So that means that round is dominant over wrinkled. In the seed color category, if we crossed yellow with green, that means that all of them in the F1 generation ended up being yellow. That indicates that yellow is dominant over green. With the pod shape inflated versus constricted, all of them are inflated. Again, inflated being dominant, green versus yellow, pod color, all green, green being dominant, and purple being dominant over white. That's all in what we call the F1 generation. What do I mean by that? I mean that this is the very first generation, so one, the first generation to come from this test cross. And as you can see, because all of them in this F1 generation of all these different crosses are going to display a single characteristic, the round over the wrinkled, for example, or yellow over green, depending on which one you're talking about, it's going to display the dominant characteristic in the entire F1 generation. But when we breed the F1 generation with itself, so when we do a cross of the F1 versus the F1, we end up with what we call the F2. So this is the second line in this generation. And at that point, we start seeing these traits come back out again, right? And we're going to actually see them, you might not necessarily recognize this mathematically, but in about a 3-to-1 ratio. So there's about 3 times as many yellows and 1 times as many greens. 3 times as many rounds as there are wrinkleds, right? 3 times as many inflated as constricted. What does that mean? That means that the recessive phenotype is coming back out in the next generation because they're going to have this independent assortment of what Mendel was calling inheritance packets. But we now know that they are alleles, which are found on chromosomes, right? Okay, so this is just showing a little bit more of the different traits crossed here if we're looking at the flower position, axial versus terminal, tall versus dwarf, etc. And all of these are dominant and recessive characteristics. So there's other ways that these genes could interact, but this is the simplest way that genes can interact. One is stronger than the other. One dominates over the other. Okay, so why did Mendel do this? What was his plan? His mathematics allowed us to check out exactly what percentage of each of these generations was going to display which phenotype. And he did this in such a really specific, meticulous way by cross-fertilizing specific pea plants with different traits to study the frequency of the phenotypes in the offspring. Now, again, he was not using what we currently use in terms of our genetics, in terms of our nomenclature. He was using terms like true breeding, which we currently would be using the word homozygous. He was also using inheritance packets when he should have been using alleles or genetics. Remember, this was all happening way before we understood or even had the ability to look at chromosomes inside the cell. So homozygous meaning the same for both, and that's what true breeding means as well. And an allele for the trait could sometimes be different, meaning that the individual could be heterozygous at that location. Picture when we're talking purple flowers, right? Purple flowers could be purple because they have 2 dominant alleles, aka homozygous, or because they have 1 dominant allele, 1 recessive allele, but the dominant allele codes for purple color, so they're heterozygous genotypically, but appear the same phenotypically as the homozygous dominant, right? So Mendel did this with a ton of different traits and started making repeated observations over and over. No matter which contrasting variety he crossed, one of the traits disappeared in the F1 generation. Why? Because the dominant allele took over the recessive allele phenotypically. And we now know that that first F1 generation are all heterozygotes. And we'll discuss that in a moment when we look at the Punnett squares. So when we take the heterozygotes, the F1 generation, and cross it with itself, that gives us the F2 generation. And at that point, he starts seeing the reappearance or the reemergence of these recessive traits. So he started calling the one expressed in the F1 generation the dominant trait and the one that disappeared the recessive traits. And then remember that recessive trait does pop back up in the F2 generation. And then I started looking mathematically and found that consistently we had the same proportion of expressed traits for each of the different crosses. It was always a 3-to-1 ratio. 3/4 expressed the dominant and 1/4 expressed the recessive. So that's the phenotypic ratio. So he determined that the phenotypic ratio, again, the outward appearance ratio, was 3-to-1. 3 out of 4 plants, 4 had purple flowers, 1 had white flowers. But that's not getting to the whole truth of the matter, because what we're actually looking at would be the genotypic ratio, where we're looking at the purple plants that are there because they have 2 dominant alleles, the purple plants that are purple because they have 1 dominant and 1 recessive allele, and of course the plants that are white, which are the homozygous recessive, and those are going to be the same. Those are the same one here. But this 3 is actually split up into one of them is going to be homozygous dominant and 2 are going to be heterozygous. And this is going to become more clear in a moment when we start doing Punnett squares. Okay, so let's just kind of walk you through this a little bit further. In the P generation, that's the parental generation. In the parental generation, we are crossing one true breeding purple. Remember, that's Mendel's words. Our words would be a homozygous dominant. But a homozygous dominant purple with a homozygous recessive, which would be the white. You'd be calling that a true breeding white. That would be the same thing. We'd be crossing a true breeding purple with a true breeding white to make that F1 generation. And in order to make the F1 generation, remember, we're actually going to be undergoing the process of meiosis. So that means that we're separating out, we're segregating. We have 2 alleles here, but only 1 is making it into the haploid gametes. So I'm going to take a second to introduce some more nomenclature, the words haploid and diploid. Okay, the parental generation, as well as the F1 generation, etc., these are diploid. They have 2 copies. But the sperm and the eggs, in this case the pollen, are going to be haploid. That means they only have 1 copy of an allele, right? And then these alleles are going to come together when the sperm and the egg come together to make the F1 generation. So only 1 allele can come down from each of their parents, allowing the fusion of these 2 gametes together to be able to make what we now know is a heterozygote, right? 1 dominant and 1 recessive allele in the F1 generation. I also want to say something here. A lot of people might ask, why isn't this WW? And that's for simplicity's sake. So when we have a dominant trait, in this case purple, purple's going to get the capital letter, capital P, and then white is going to get the lowercase letter that matches the dominant trait. So if we're looking at yellow versus green, capital Y, lowercase y, because yellow would be dominant, right? And so depending on how you're looking at it, this way we can recognize that these 2 traits go hand in hand, except that one is dominant over the other. Okay, so these 2 alleles, All right, so now let's take a look what happens when we cross that F1 generation. This is called self-fertilization. Here we have the heterozygote, has 1 dominant and 1 recessive. This one also has 1 dominant and 1 recessive. And when we cross these out together, this can segregate into here and here. Remember, we're going to be putting them, the alleles are segregating out separately. Each of the gametes can only bring 1. Here we have the capital P, and this one's lowercase is going to go here. So now we're looking at this guy here, again, 1 capital P here, 1 lowercase p here. This is called a Punnett square. So please get familiar with this. We will be drawing these out. You will see these on exams. A Punnett square is a way that we can look at the way the F2 generation sorts out in a cross, or really how any generation sorts out in a cross. Knowing the parental phenotypes, we can guess the probability of the offspring. Namely, that if we were to cross these 2 together, we will end up with, we're going to come pull this one down and that one down. This one's going to be a homozygous dominant. It has 2 capital P's. This one here, 1 capital P, 1 lowercase p. It's a heterozygote. Still going to have the dominant phenotype, but is a heterozygote genotypically. Same thing with this one, 1 capital P, 1 lowercase p, heterozygote genotypically. So 1 to 2 to 1 here. This guy here has a 2 lowercase p's. Is going to express as white. Okay, so we have a 3-to-1 phenotypic or outward appearance ratio, but a 1 to 2 to 1 genotypic ratio, right? 1 to 2 to 1 genotypic ratio. Okay. So these are the way that we're looking at the genotype versus the phenotype. Again, genotype is going to be what happens with the genetics. Are we homozygous dominant? Are we heterozygous? Are we homozygous recessive? But the phenotype is going to be the outward appearance of the individual. Purple purple, or white. So this is what I mean when I say the genotypic ratios and the phenotypic ratios might not line up. The genotypic ratio is going to be 1 to 2 to 1, and the phenotypic ratio is going to be 3 to 1. Does that follow? All right, but again, this is really just probability. If we only have 4 plants, we're not necessarily guaranteed that it's going to be split up that way. The thing that is beautiful about Mendelian genetics, or really about Mendel's work, is that he investigated such a high number of these plants that he got really tight, what we call N, or the number of the statistical number of the different plants that we use, right? And if we get a high enough N, the probability starts to become very clear. So the probability means the likelihood that an event will occur. If we flip a coin, it can either be landing on heads or tails. If we only flip a coin once, it is 100% either heads or tails. If we flip a coin twice, chances are it could be 50-50, but there's no guarantee of that. It could be 2 heads in a row. If we flip a coin 1,000 times, it's very likely that it's going to flip 499 one way and 501 the other. So the higher the N, the higher the statistical probability that you're going to get accurate data. Okay? So again, probability refers to the likelihood that an event will occur. So if we're going to flip 2 coins, there's 4 possible outcomes. We can either have both of them on their heads, both of them on their tails, one on their head and one on their tail, or the other one on their tail and the other one on their head, right? 4 possible statistical outcomes. So we have probability tree here. We have 50-50. We're going to flip this one and then flip that one, right? We flip the coin twice. It can either be heads and then tails, or heads and heads. Sorry, this would be tails and then tails, tails and then heads, heads and then tails, heads and then heads. So 4 different statistical probabilities. All of them will be a quarter chance. Now, if we flipped a coin 4 times, we probably would not get one of each of these results, right? So if we flipped 2 coins 2 times, we would probably not get each of these results. But if we flipped it 1,000,000 times, we would probably get it a quarter of the time, a quarter of the time, a quarter of the time, and a quarter of the time, because the higher the N, the better the chances of showing us the exact statistics. And that was the brilliance of Mendel, is that he used a really high number of these plants to be able to come up with really tight statistical analyses. All right, so a Punnett square is basically a way that we can look at the probability, the possible gametes that are going to be created from one individual gets put on one side, the possible gametes that are able to be created from the other individual is put on the opposite side. And then we pull them together down into their different boxes, and we look at the genotypes of the potential offspring inside the square. And we just showed you that for what we call a monohybrid cross, which is when we are looking at one trait at a time. And this is probably all I will expect from you for the purposes of this class, is a monohybrid cross, but at the end of this lecture, we're also going to walk you through a dihybrid cross. Let me say that again. A dihybrid cross, which is when we are going to have 2 traits that we are looking at the same time. All right, so let's walk you through some of the mathematics here. If 2 pea plants are heterozygous for flower color, that means they're half purple and half white in terms of their alleles. That's their genotype. What would their phenotype be? What color would they be? And that answer would be purple, right? The heterozygote appears to be purple. What are the possible gametes that they could form? Well, they have one capital P and one lowercase p, right? One dominant and one recessive. So they could form 50-50, capital P and lowercase p. What offspring could they produce? Well, that depends on what they're crossed with, right? I'm assuming that we're saying we're crossing them with another one of the heterozygotes, in which case we have 3 different options. We can have the heterozygote, and that'll be half of them will be the heterozygote. That's the 2 in the 1-2 to 1 ratio. And then we'll have 25% will be the homozygous dominant, and 25% will be the homozygous recessive. Again, those ones in that 1-2 to 1 ratio. And the probability that pea plants would have offspring with white flowers is only 1 out of the 4, right? 3 are going to be purple, 1 is going to be white. Okay, so here's our answers. Purple, capital P or lowercase p. Depending on how you cross it, we would be getting the homozygous dominant, the heterozygous, or the homozygous recessive, meaning the offspring could be either purple or white at a ratio of 75% purple and 25% white. Now let's walk you through this if you're still kind of having trouble understanding what I was talking about here. From that F1 generation, remember everybody's going to be a heterozygote, so that means everybody is going to be one capital P and one lowercase p. And if we're going to separate that out, capital P here, lowercase p here, again we're separating this one, capital P, lowercase p. If we were to pull them down we're going to get these guys here will be the 1/4 that are going to be dominant, homozygous dominant, both with capital P's. Then we pull this one down and that one down. Here we're going to get a heterozygote. There we're also going to get a heterozygote, so that's going to give us 2, right? So this is a 1 to 2, and then this last guy here is going to be 1 ratio. So what do I mean by 1 to 2 to 1? One of them is going to be homozygous dominant. One of them is going to be homozygous recessive, appearing as white. 2 of them are going to be heterozygotes. That's a 1 to 2 to 1 ratio of the genotype. But again, if we're talking about the phenotype, we're looking at a 3 to 1 ratio. Okay, so I hope that clarifies for you. This is this table is just a series of multiple dominant recessive human traits. You can go ahead and take a look at them. One of the ones that we use quite often in class here is the earlobes, whether or not they're free or attached. And if you just go ahead and look at your family tree, you can see immediately where you get what traits from. If you're just looking back at your family line and some photographs and start looking at some of the things that you may express that other people in your family may or may not express, you can see which ones come down the line from where. Okay, so let's talk about the difference between the homozygous dominant and the heterozygotes, because phenotypically they look the same, right? So how would you be able to tell if you had a purple plant, or a plant with purple flowers rather, if it was a homozygous dominant, remember Mendel called that a true breeder, or a heterozygote? And so what do? Well, we cross it with the homozygous recessive. And guess what? We always know what the homozygous recessive is because that one is the white one. It has a nice different phenotype. So it has a different outward appearance than the others. So you can tell for sure. So its phenotype is going to match its genotype. So if the unknown is homozygous, then we're going to have all of the offspring expressing the dominant trait. But if it's heterozygous, then we'll get 50-50. What do you mean by that? Well, let's take a look. Okay, so if we cross the purple plants with a true breeding white plant, so this is going to be a heterozygote plant. If it's a heterozygote, that means that we're going to be able to create the capital P or the lowercase p when we're making the gametes. The white is only going to be able to create the lowercase p. So instead of making the full square of 4, we've just made a 50-50 here, which indicates here that 50% of them are going to be the heterozygote, and 50% of them are going to be the homozygous recessive. That would indicate, if we got a 50-50 ratio or a 1-to-1 ratio, that we had an F1 purple plant that was a heterozygote. If, however, we had an F1 purple plant that was what he called a true breeding purple plant, right, a homozygous dominant, it's only able to make the capital P. This one's only able to make the lowercase p. 100% of the offspring are going to appear as the heterozygote with a purple phenotype. So this is how Mendel was able to determine which of his plants were true breeding so that he could use them for future crosses. And this is what he called, again, a test cross. Okay, so Mendel then started looking at, again, he was talking about them as inheritance packets, but he started looking at how they traveled together. Did they travel independently, or how did they assort? And so he was looking at the alleles of a trait and seeing how they separate from each other during the formation of gametes. And basically what happens is one half of the gametes carry one copy and the other half of the gametes carry the other copy. And that's because that just has to do with the way meiosis works, right? The way the diploid germ cells form haploid gametes. So which chromosome each of them segregate into is completely random. And what that means is that when we are separating out chromosome 1, maternal and paternal could go left and right, or right and left. And for chromosome 2, maternal and paternal could go left and right, or right and left. Do you see what I mean? Over and over and over, all the way out for 23 pairs, which gives us over 2 billion possibilities of combinations just from independent assortment alone. Right? And that's not including anything that happens from what we call crossing over, which we can talk about in a little bit when we get to meiosis. But crossing over is basically a shuffling of the genes that happens right before that independent assortment. Okay, so now let's hop into the dihybrid cross a little bit. If the parent P's that we're starting with have, and it says genotypes here, why? Because we're looking at 2 different traits. Round and yellow are dominant over wrinkled and green. Okay, so if we're looking here, we have dominant, but we have a heterozygote for the Y here, which is going to be yellow. And we have heterozygote and recessive here. So question before we even start, what is the expression here? Well, it's going to be round because we have two dominant, and it's going to be yellow because we have a heterozygote here. This one is going to be round because it's heterozygote, and it's going to be green because it's going to be homozygous recessive. Okay, what are the possible gametes that we can make? Well, now we get a little bit complicated because each of these is going to make its own gametes. Now, when I say gametes, remember the R is going to separate independently from each other and the Y's are going to separate independently from each other, but they're still going to end up with one R and one Y in each of them. So for this side, we can have capital R and capital Y. We can have capital R and lowercase y. We only have a 50-50 chance there because we're only going to be able to get the capital R. In this case, we can make capital R and lowercase y or lowercase r and lowercase y. Okay? And we can cross that out for you in a moment. So how are we going to find the possible gametes? Well, using FOIL. First, outer, inner, last. What do I mean? First, outer, so this is the first one, R and R, right? Then we have outer, R and Y, right? And then inner, right? So then we're going to have, so you can cross each of these together, right? So we have 4 different combinations of how we're going to be able to pull them down. It'll be easier for me to show you in the next slide. So anyway, use FOIL, first, inner, outer, last, and then we get to these particular gametes. So that's going to be the capital R, capital Y, capital R, lowercase y, lowercase r, upper Y, lowercase r, capital Y. So those are the options that we're going to be able to have for our possible gametes. All right, so in this case, we're going to be given the cross. So here we have the F1 by F1, which means that they're each heterozygotes for both traits. Capital R, lowercase r, capital Y, lowercase y. Again, heterozygote for both of them, capital R, lowercase r, capital Y, lowercase y. That means that when they can make their gametes, each of them can make 4 different combinations, both capital R, lowercase y, lower r, capital Y, and both lower. And same thing across here. And then when we pull these down, we're going to do the same thing. It's just a little more complicated. We're pulling both R's together and both Y's together. In this case, we are going to have all dominant alleles. Here we have the lowercase r, lowercase y, lowercase y. Here we are going to have all recessive alleles. So 1/16 are going to be homozygous dominant for both, and 1/16 are going to be homozygous recessive for both. But the rest of them are going to be a combination of that, right? So we're going to have many different combinations of the R's and the Y's that allow us to become yellow and round. Why? Because yellow and round are our dominant phenotype. So we only need one copy. So here we have one capital R and one lowercase r, but it still appears round, right? Here we have heterozygous for both. It still appears round and yellow. So the only way that we can get those recessive phenotypes to come out is when we start seeing here is the lowercase r's, that's going to give us the wrinkles. So until we start seeing this homozygous recessive, we're not going to see those recessive traits. But what I want to point out here is that the ratio of these, the way these are going to show up is going to be 9 to 3 to 3 to 1. 9 of them are going to have both dominant phenotypes, in this case round and yellow. 3 of them are going to have one dominant phenotype, in this case round, and one recessive phenotype, yellow, right, because it's green. So green is going to be the recessive phenotype. Here, we're going to have one recessive phenotype because of the R's are both low or lowercase. So that's going to be homozygous recessive, meaning that they're going to be wrinkles. And we're going to have the other dominant phenotype, which is yellow. And that last one, so again, 9 of these guys, 3 of those guys, 3 of these guys, and 1. 9 to 3 to 3 to 1. And this last one is going to show both of the recessive phenotypes. So that's how dihybrid cross works, guys. And don't worry, we probably won't have to walk you through that again. I just want you to kind of understand that these traits, if they're both on this, if they're on independent chromosomes, they're going to independently assort, which means that we're not just looking at one trait at a time, where we as the individual are going to be looking at multiple traits at a time. Okay, I also want to tell you that Mendel looked at the simplest way that these traits could interact. Dominant and recessive is the easiest version of how traits interact. Oftentimes, the way a phenotype is expressed is not straightforward. Traits can have a lot of different outward appearances when what's called polygenic inheritance occurs, when multiple genes act together. A good example of this is human height, human skin color. There's multiple different genes that play a role, and so it's not just one. And we also can have what's called pleiotropic inheritance, where a single gene can cause multiple different phenotypes. So different changes in the particular gene that causes cystic fibrosis. Again, that's called pleiotropic inheritance. All right, so here's polygenic inheritance. Polygenic inheritance is going to be when we're looking at, for example, the height of individuals. Here's a bell curve showing, here's the idealized bell curve showing the shortest, the medium, the average height to the tallest. But then what we're actually seeing is a range of heights here in the distribution of individuals. So the actual data doesn't always match the prettiest data, but the point is that this is going to have multiple different genes that affect it. So we're not just going to have tall and short, we're going to have every range in between, because we have everything from multiple short genes over here to, from multiple short genes over here to an equal amount of short and tall genes to multiple tall genes over there, because they're all going to be expressed and all interact to cause this phenotype. So a lot of things are going to interact in a way that we call non-Mendelian inheritance. So again, Mendelian inheritance is going to be just very simple, dominant recessive characteristics. But a lot of things are polygenic, like height, weight, skin color, and intelligence. And a lot of things have epigenetics, or what we call environmental factors, that can influence them as well. So epigenetics is kind of like the the nature part of how your genes are going to interact with you. And again, most diseases are also polygenic, like cancer, diabetes, schizophrenia, etc. We also can have incomplete dominance. So again, fully dominant and fully recessive is kind of the prettiest version of how this can work out. But sometimes heterozygotes can have what's called incomplete dominance, where we have an actual intermediate phenotype where the heterozygote shows a blend between both of those. Good example is, instead of, remember we talked with purple and white, and purple was always going to be, even the heterozygote was going to show up purple. But with red and white, say we get a pink, a heterozygote that's kind of a blend of both, right? An intermediate phenotype. Another example of this is curly hair. So people with straight hair who mate with people who have curly hair end up with a heterozygote that is going to be wavy hair. So when the heterozygote has a completely independent phenotype of either of the dominant or recessive homozygote individuals. All right, so here's incomplete dominance. Here's going to be if we have two of the dark of red colors, that's going to give us red snapdragons. If we have the white, two of the whites, going to give us white snapdragons. If we have one white and one red, we're going to get pink snapdragons. So previously we saw that the genotypic ratios and the phenotypic ratios did not line up, but with incomplete dominance, they do. With the incomplete dominance, we get a 1 to 2 to 1 ratio, which matches exactly, so that's the genotypic ratio, and that matches exactly with the phenotypic ratio. What I mean is that the phenotype of the heterozygote is distinct from the homozygous dominant and from the homozygous recessive. Okay, so as I mentioned previously, in an individual, you may have two copies of whatever the gene is. They can both be the same or they can be different, because we have one maternal copy and one paternal copy of each chromosome. However, even though we can only have two in our body, there can be many within a population. So there can be much more than two alleles within a population. And so sometimes we have many alleles that are going to interact, and sometimes there's not necessarily a dominant allele. We can end up with expression of both. Right? That's going to be something that's said to be codominant, like when we have red and white heterozygotes, okay, like we just spoke about. But let's expand that codominance to human blood types. So we have blood types A and B and O, right? So we have a gene that determines the A and B blood type, which exhibits more than one allele. It has it can have either the A allele, the B allele, or the O allele. Now, we as the individual can only have two, right? We can either have, we can have any combination of two. Now, if we get the gene that encodes A or B, it's going to be encoding an enzyme that adds sugars on the surface of the cells. Okay, so if we get the A and B marker, we're going to, A or B marker, we're going to create the A or the B sugar. So here's the gene that encodes the enzyme. It's designated I. It has three alleles, IA, coding for the A sugar, IB, coding for the B sugar, and lowercase I, coding for no sugar. What does that mean? That means that A and B interact as partners, right? They kind of battle it out with the strength, because they're going to be, hey, I want to make this one and I want to make that one. So if we have the A and the B, we're going to make both. But with the I, we are making nothing. So that means that the I is recessive to both A or B. So A and B exhibit codominance, and A over I or B over I exhibits dominant recessive. So different combinations of the alleles can produce four different phenotypes, four different blood types. We can have A, B, AB, or O. In order to have the O phenotype, you have to have two copies of the lowercase I. In order to have the AB phenotype, you have to have one copy of A or one copy of B. But in order to have A, you only need to have one copy of IA. So you can have IA and lowercase I, or you can have two copies of IA. And same thing for B. You could have an IB and lowercase I, or you can have two IBs. Okay? So again, both A and B are dominant over I, but A and B are codominant with each other. So this makes it really complicated when we talk about how we can donate blood types, because we have antigens, which we were just talking about the sugar. So type A makes the A sugar, type B makes the B sugar, type AB makes both sugars, and type A makes no sugars. But the opposite of these sugars or antigens are what we call antibodies. Antibodies attack antigens, so we can't have them in the same location. So if we have the A antigen, we cannot expose it to the A antibody or we will clot. If we have the B antigen, we cannot expose it to the B antibody or we were clotting. So if we're expressing the B antigen, we do not express the B antibody, but we express the A antibody. If we're expressing the A antigen, we do not express the A antibody, but we express the B antibody. But if we're expressing both antigens, both sugars, we're not going to make antibodies to either because we don't want any clotting. And if we're type O, we've got no antigens to worry about. We can make antibodies to both, no problem. But that means that O is not able to accept any blood from anybody that has any antigens because it has antibodies against both, right? That means that type A can accept it from anyone because it has no antibodies at all, so it can accept any type of blood. So AB is considered the universal acceptor, and type O is considered the universal donor. Okay, so here's a table that shows you how you can link the genotype and the phenotype. If we are type A, we can, if phenotypically type A, you can be either of these genotypes. You can be homozygous dominant for A, right? So, or you can be heterozygous with A and lowercase I, or what we would consider to be O. Type B, same thing. You can have two copies of the B allele or one copy of the B and one copy of the I. Type AB, you're heterozygote for A and B, but they're codominant, so they're fighting each other out. And type O, you're homozygous recessive for the I. So I know this is kind of complicated because we're talking about how they may interact in terms of being codominant or whether they're dominant recessive, and what kind of expressions we're going to have. Plus, we're also talking about the sugars that are on the surface of the cells versus the antibodies that are circulating through the bloodstream. All right, so let's look at the alleles that could be coming from the gametes from the mother or the gametes from the father, okay? So if we have, these are all three of the gametes that you could possibly have. So when it says from the father, that doesn't mean that the father has three alleles or that the mother has three alleles. It just means that any mother from the entire population could donate any of these three alleles. So just to be clear, previously when I said that you can only have two, that's true. You can only have two, but there are three options within the population for expression, right? A, B, or O, which is the lowercase I. If we get two copies of the A allele, we're going to demonstrate to be A. If we get one A and one B, we are AB, and if we get the A and the I, we express as A. If we have the B and the A, we're going to express as AB, same as here. If we have the B and the B, we're going to express as B. If we have the B and the I, we're going to express as B. If we have, again, the A and the I, we're going to express as A. If we have the B and the I, we're going to express as B. And if we have the I and the I, we're going to express as O, right? So that is the way that these interact in terms of the genotypes and the expressions of the phenotype. Okay, so let's talk about epistasis for a second. So epistasis is going to mean that the expression of the genes are dependent on other genes that are upstream or sometimes on other chromosomes entirely that are called modifier genes. Genes that are going to increase or decrease the expression of the target gene of interest. And epistatic mutations are kind of interesting because they can have multiple effects that are when we combine them together that are separate from each of the genes individually. Right, so here's epistasis, for example. Here's a black Labrador, a yellow Labrador, and a chocolate brown Labrador. Okay, so depending on how the gametes are going to sort out, we can end up with different phenotypes. So here we have homozygous parents, We're going to take the homozygous black and the homozygous yellow. We're going to mate them together. And the F1 puppies are all going to be black, that they express capital B, lowercase b, capital E, lowercase e. And then when we express them all back out together, if we end up with the ability to have lowercase b's and any expression of E, we end up with chocolate lab. So this is kind of like the 9 to 3 to 1 that we saw previously, right? 9 to 3 to 3, except this 1 is also going to express as the yellow lab, right? So it's very similar to that in that we have an actual separate phenotypic appearance when we have the expression of the dominant E but the recessive B. So that's how we're going to get that beautiful chocolate brown lab phenotype. Okay, we can also have a bunch of epigenetics or environmental factors that play a role in the expression of alleles. So the degree to which alleles are expressed depends on the environment. Some alleles are heat sensitive. So for example, the Siamese cats, or again, Arctic foxes, they're going to produce pigments only when temperatures are exactly right. So the Arctic foxes produce pigment when the temperatures are warm, which means that when it is cold, they're going to be white, which makes sense because they're hunting in the snow. When it's warm, they're going to be a little bit darker, so they're going to look like the dirt and the rocks that are around at that point. Siamese cats are going to produce fur pigment only when the temperatures are colder than their body temperature. So the rest of their body, the main part of their body is going to be white, but their ears and their paws and the ends of their tails are going to be darker because these are going to be the areas of their body where the blood cools a little bit. Another environmental factor that's an example is hydrangea flowers. We can change the color of the hydrangea flowers from blue to pink depending on the acidity of the soil. So people will put things into the soil to try to get the flowers to bloom whatever color it is that they prefer, or change the soil content so they can get blue and purple throughout their entire hedge. Right, so here's some examples of what I mean by that. Here's hydrangea flowers. Depending on the acidity of the soil, they're going to be either blue or like a red pink. Here's that same Arctic fox in the winter when temperatures are cold, and in the summer when temperatures are warm. Again, you can see it blends in better in both of those environments. Okay, so some other examples of non-Mendelian inheritance include sex-linked genes. So here's a good example of sex-linked genes. A male fruit fly is known as Drosophila over here, and it's commonly studied in scientific research laboratories because it has such a quick lifespan that you can study multiple generations within the a thesis term. So a male fly was discovered with white eyes instead of the typical red eyes. Here's what the regular eyes look like, but he had white eyes. Why did the mutated white eyes occur mostly in males only? How is that possible? And that's because it's determined that the white eye trait is actually found on the X chromosome, which means that in order for it to be expressed, it's going to be expressed more in males because males have one X and one Y. They're what we call hemizygous. That means that they're going to express from that X whatever that X has because they don't have another X to express it. And so if the white eye allele is found on the X chromosome, it's going to express white. But with females, because they have two X's, you might have one X that has the white, but the other one probably has the red. And so we're not going to express the white, we're going to express the red. And again, just like with humans, XX is going to be female and XY is going to be male. Good examples of sex-linked traits are going to include hemophilia, for example, or male pattern baldness or color blindness. These are all traits that are expressed on the X chromosome, which means that they're going to be more likely to be expressed in males because, again, a hemizygous male who has a mutation is only going to be able to express what his X actually presents, whereas a female who has two copies, if one has a mutation, they might be able to use the information from the other X instead. Okay, so here's how X-linked inheritance basically works. Here we have the male with the red eyes and the Y, and the female with the red eyes, XR and X white, W being the lowercase. I mean, I guess it should be a lowercase r, but they did it that way just to show you it's going to make white. So what happens? So that female is able to make either this X or that X in terms of her gametes. So this one's going to have regular red eyes, that one's going to have the white eye gene. Now this one is a male, he's got an X and a Y. Only this is whatever's on that X is going to be expressed because the Y can't express anything. But when we're making gametes, he's going to make that XR, that the regular red eye gene. So when this comes together, here we end up with a female XX that has two red genes, who's going to be a female with red eyes. Here we have XX as well, It's a female with one regular red eye and one white eye gene, but that means it's going to express red eyes because we have the dominant allele there to be able to act in a dominant recessive fashion. Here, this male is going to get the Y and the XR. So we have an XY, that's how we know it's a male, but the R is going to indicate red gene or red eyes, so we have normal red eye phenotype. This one, on the other hand, is going to get the Y and the XW, indicating that it's a hemizygous. I mean, these are both hemizygous for the X, and it's going to express whatever the X has, in this case, white eyes. So that's why the males are going to present with white eyes more often than the females, because in order for the females to present with white eyes, they need 2 bad copies, not just one. Again, other examples of X-linked genes include color blindness, hemophilia, and muscular dystrophy. Now, we were just talking about how things independently assort, but not all genes independently assort. Some genes are found on the same chromosome, and if you're found on the same chromosome, you tend to segregate together, particularly if you're found very closely linked on the same chromosome. Now, the further apart the genes are on the chromosome, the less likely they are to be linked, because there's a higher likelihood that crossing over or recombination will occur between them. So if they're on the very tips of the chromosomes, they're probably going to act as though they're on totally different chromosomes. But if they're really close together, the chances of crossing over happening between them is smaller. And so depending on how close they are, the closer the 2 genes are on the same chromosome, the less likely they are to have crossing over separate them, the more likely they are to be what we call linked and travel together. What do I mean by that? Okay, so here is a series of sister chromatids and a homologous pair. So a homologous pair is going to have the maternal and the paternal pair, right? And here again, we're going to have sister chromatids that have the exact same genes, because these are copies of each other. These guys with the homologous pair are going to have this, the exact same alleles from the sister chromatids, but they don't have to be the same alleles among the 2 homologous pairs. So even though we're looking at, say, eye color, this one might have blue eye color for both of these and brown eye color for both of those. We might be looking at height, and this might be, I don't know, really, really tall and really, really short or whatever. Lots of different traits that might be expressed, but they're going to be expressing differently depending if you're looking at blue eyes or brown eyes, white, right? But when we have the meiotic event occurring, when we are creating sperm and eggs, we have something called recombination or crossing over occurring. Basically, this arm lies over that arm, and they swap, right? And so, as you might imagine, the 2 genes A and B are closely linked because they're closer together. It's less likely for the cut to happen here. It has to happen anywhere from here to there than it is for the cut to happen between this one and that one. It has to happen anywhere between here and there. So it's much more likely for it to happen between A and C than it is between A and B, right? Because of how closely these 2 are related. And when crossing over occurs, now they're going to be independently assorting again. As I mentioned, we normally have 46 chromosomes. That's going to be 23 pairs. 22 of them are autosomes, meaning that they are non-sex chromosomes. 1 pair is the sex chromosomes, either XX or XY, and that determines your sex. Yes, females are XX, males are XY. But all of the genes on the X chromosome, all the genes on the Y chromosome, all the genes on the Y chromosome are dedicated to male differentiation and development cascades. So there's not much on the Y that matches up with the X, except what's called the pseudo-autosomal region that allows them to bind. But most of that is just going to be, most of the Y is just going to be turning on male differentiation and development. So as the chromosomes are separating out during meiosis 1 or meiosis 2, we're it's supposed to normally end up that we are separating them in half and then in half again. But every once in a while, alleles don't separate out properly, and that's called non-disjunction. That leads to what's called aneuploidy, and aneuploidy means abnormal chromosomal number, and it's really not very well tolerated. Most of these aneuploidies are going to cause early embryonic death or failure in fetal development. Typically, this will be a miscarriage event. However, there are some aneuploidies that are tolerated only as triploids, so as an extra copy, not as a monoploid, which would be one less copy. But if we do end up with a triplicate copy of chromosome 21, sometimes 22, sometimes also 18, sometimes also 13, we can end up with chromosomes that are extra. Neither of those, 18 and 13, are not going to be, 8 and 13 are not going to be anyone that are going to survive to adulthood. But 21 and 22 are able to survive to adulthood with severe developmental delay and mental retardation, as well as having adducted thumbs and some other characteristics. And that triploid, or third copy of chromosome 1, is characterized by a syndrome known as Down syndrome. Okay, and how does that happen? Well, non-disjunction basically happens. You can see what should have happened here, right? Is that one of them should have come this way, and one of them should have gone that way, but instead both went that way. After they both go this way, now we're going to make a separate set, but now we end up with 3 here instead of 2, and 1 here instead of 2. This is called monoploidy, and it is not tolerated. This is lethal. Missing chromosome N-1, death. Extra chromosome N+1, not super well tolerated unless it happens in chromosome 21, or again 8 and 13 and 22, but again, those are not going to be very well tolerated. Okay, so here's some examples of non-disjunctions that are tolerated. Trisomy X is having 3 copies of the X. Why is that well tolerated? Because of something called X inactivation. So normal women who have 2 copies of X are going to inactivate 1 so that only one of them is useful at any time. And a woman who has trisomy X is going to inactivate 2 of them. For some reason, the X inactivation just goes, instead of saying I'm inactivating 1, it says I'm going to inactivate all but 1. However, it is characterized by a phenotype of a very tall stature and some learning disabilities, as well as limited fertility, so some infertility problems. We also have XO, which is known as Turner syndrome. So instead of getting an X or a Y as the second sex chromosome, they just get 1. But what happens with this, again, thanks to the way that X inactivation works, it inactivates all but 1, so we just don't have any inactivation here, and this individual is going to end up okay. However, it's going to end up with immature sex organs and infertility issues. This individual will express as female and will be a little bit shorter in stature. And you see that the X chromosome somehow is a little bit linked to height, right? Because if we end up with an extra one, we're tall, and we end up with less one, we're short. We also have XXY. XXY is known as Klinefelter syndrome. Klinefelter syndrome is where we're going to have expression of a little bit of male and female characteristics. We're going to be expressing as Y because remember, as male, sorry, because remember the Y is going to cause the sexual differentiation and development of the male, so it's going to cause that whole cascade. But we're also going to have breast development occurring because the X is going to, 2X are going to start inducing this. We're going to have some infertility and perhaps a little bit of hermaphrodism, right? A little breast development, smaller gonads, so a little bit of ambiguity in the sex characteristics. And then also here we have XYY. So 2 Ys seems to be pretty well tolerated. Why is that? Why is that though? Because Y only starts the male differentiation and development cascade. It's really only needed during embryogenesis. So it's turned on during embryogenesis and then it's shut down basically, not really utilized again. Except during meiosis or making sperm. So this is really going to be fairly well tolerated. However, it has been demonstrated that there is a higher percentage of this XYY in prison populations and aggressive and violent offender populations than in the normal male populations and the outside of the prisons. And so it's possible that that extra Y is indicative of higher levels of testosterone and perhaps aggression. All right, so anyway, those are non-disjunction disorders. Now, let's talk about how mutations can change what gets passed down from one generation to another. So any mutation that occurs in the somatic cells, which are the cells that are not your sperm and egg cells, are not going to be passed down to offspring. But any mutations that occur in the germ cells, which are going to be your sperm and your eggs, can be passed down into future generations. And pretty much anything that's a hereditary mutation that does not cause death is going to almost always result in recessive alleles. And remember, recessive alleles can be maintained in the population because they're not expressed in most individuals, right? The heterozygotes are going to have the dominant phenotype, so they're going to be able to have no problem. It's only going to be the recessive individuals that are going to end up with these really harmful genetic disorders, right? So in some cases, mutant alleles become more common in human populations, and then when we cross them together, we end up with a recessive individual who has a genetic disorder. Autosomal recessive disorders include things like sickle cell anemia and Tay-Sachs. Sickle cell anemia is an autosomal recessive disease where the hemoglobin gene is mutated. Now, hemoglobin is what carries oxygen to your cells, so that's going to cause odd red blood cell shapes and reduce oxygen carrying capacity. Tay-Sachs is another autosomal recessive disease. It's a disease where a gene that codes for a lysosomal enzyme is mutated, and it can cause issues with brain deterioration, and usually this is going to lead to death very often before puberty and adolescence hits. Here's sickle cell anemia. Sickle cell anemia is caused by, again, a mutation in the hemoglobin gene. It's called sickle cell anemia because this literally looks like a sickle, right? Like a scythe that you might use to chop down grass. And as you might imagine, the oxygen carrying capacity of this particular cell is drastically reduced compared to the normal red blood cell. Another autosomal recessive disorder is cystic fibrosis. There's a mutation in a chloride channel gene, which causes a disruption in the water balance inside of their cells, which means that they have too much mucus produced. Increased mucus is going to impact lung function as well as liver function, pancreas function, etc. And here's another example of a disorder, but this one is going to be a dominant disease. So for all of the other ones, the recessive diseases, you have to have 2 bad copies. But for a dominant disease, you only have to have 1 bad copy. A good example of that is Huntington's disease. We have CAG nucleotide repeats that get inserted into a gene, and depending on how many there are, we're going to end up with a disease state. More than 35 causes the disease, and the more repeats you have, the earlier the onset of this brain deterioration. And the brain deterioration usually starts sometimes in their early 30s, but what's the kicker here is that usually people in their late 30s, mid to late 30s, have already had their children. So you start feeling sick and having the onset after you've already potentially passed it on to your young. So individuals who have parents who suffer from Huntington's disease often struggle with the issue of having their own offspring because they don't actually know if they're carrying this autosomal dominant disorder. But they can go and get it tested now that we have the advance of modern genetics. All right, so this here is showing the impact of cystic fibrosis. Again, it's an autosomal recessive disorder. It's caused by a chloride channel mutation called CFTR. Again, that stands for cystic fibrosis transmembrane conductance regulator gene. And it affects the respiratory system, the heart, the bones, the digestive system, the reproductive system, and the sinuses. So it's obviously going to be a very drastic impact on the individuals affected. Other examples of genetic disorders include hemophilia. It's recessive and it's X-linked. And it's going to affect blood clotting. Basically what happens is small cuts can kill these individuals because they're lacking certain clotting factors, which leads to an inability to stop bleeding. It's been famously studied in the royal family line of England, and it's kind of interesting that only the royals could possibly survive with a disease like this because an individual who was more of a pauper would probably end up cutting themselves much earlier in life. Okay, we also have another recessive X-linked disorder here called muscular dystrophy, Duchenne muscular dystrophy. It's going to be when a muscle protein that's called dystrophin. Dystrophin is going to hold your myofibrils in your muscles together, and without this functioning protein, the muscles just start to degrade and fall apart. And so it starts to affect children in childhood, and usually by age 10 or 11, they're going to be entirely wheelchair bound. So it's incredibly sad, and there's a lot of research involved in how to maintain the muscle function for as long as possible for these individuals. As always, thank you so very much for joining our class today. I really appreciate your time. Happy studying and have a great day.